Hello, this is Ming-Li Wang. I'm from Hawaii Agriculture Research Center. I would like to talk to you about our effort to bring coffee leaf rust resistance into the coffee cultivars that we already grown here in Hawaii. The work I'm going to talk about is actually from two different projects. A bigger project that's funded by USDA Specialty Crop Research Initiative. It covered most of the work. In this project, we at HARC will produce coffee seedling from crosses between CLR-resistant cultivar and Hawaiian commercial cultivars. And we will send leaf samples to Dr. Lisa Keith at PBARC, and she will then screen them to see if they are coffee leaf rust resistant or not. We will also send leaf sample to Dapeng Zhang in Beltsville, USDA ARS, and Dr. Qing-Yi Yu in PBARC, and they will do the DNA typing of these seedlings. And once the plants are planted in the field, Dr. Tracy Matsumoto will compare the performances of these plants, and including the cupping quality. And Andrea will then relay any information produced from this project to all of you guys. A second smaller project is a HARC internal project in which we will try to combine CLR resistance to nematode resistance. HARC's coffee breeding program was actually initiated by Dr. Chifumi Nagai in the late 1990s. So over the years, she imported several cultivars that carry CLR resistance. For example, Cultivar, and we actually have two different selections of Cultivar, T5175 and T86667. A second one is Obata. Cultivar and Obata, the resistance were derived from two different Timor hybrids. Cultivar came from A32/1 and Obata came from A32/2. And Dr. Chifumi Nagai had already made lots of crosses between these two cultivars and many different Hawaiian commercial cultivars. And she even advanced one of those children, F1, you can call it the children generation from the two parents. And then for one of the population, she advanced for another generation to the grandchildren or F2 population. And that particular cross was T5175 cross with MA2, which is a mocha hybrid. Recently, in HARC's Maunawili station, we identified two trees that never show symptoms. Never show CLR symptoms when they're surrounded by infected plants. And after some digging of information and some PCR DNA tests, we are convinced these two plants carry the CLR resistant gene originally identified in Liberica coffee. So these are the three sources of CLR resistance we already have in Hawaii. We understand we're kind of behind on CLR resistant breeding compared to the rest of the coffee world. There are already CLR resistant cultivars out there. However, the negotiation for intellectual property right, the importation and the quarantine procedures are complicated. We think it's still very important that we work with the genetic resources we already have here in Hawaii, and also pay attention to maintain the unique quality of the Hawaiian coffee. For Cultivar, it's notoriously known for its bad taste. So we think at the first generation, cultivar probably would bring down the quality of the commercial varieties. So we think, however, at the next generation, the grandchildren generation or F2 generation, because every individual is different, it is possible to identify individual trees that are resistant, also maintain the good Hawaiian commercial coffee quality. For Obata by itself, it tastes pretty good, and we have confidence that it's possible we can identify individual just from one generation in the F1 or the children generation. So for BA10, we missed the flower last year, so we didn't make any crosses this year. So today I'm going to focus on the work we've done in Katimora and Obata. So as I mentioned, we think we'll be able to identify good trees. Good means high quality and also CLR resistant from the grandchildren generation of Katimora crosses. And this is the F2. The first line is the F2 generation I mentioned earlier, but I'm going to talk about that later. I'm going to focus on the other crosses. We selected about 15 different crosses. They are all crossed between either T5175 with MA1. MA stand for Maui. So this particular cultivar was originally identified on Maui. It happened to be mocha type. And also OA stand for Oahu. We also have plants identified from Kauai. Ka stand for Kauai. Ka19 happened to be a red Katrai type. We also have plants identified in Kona. KO34 and KO32, both are Typica type. We also including some crosses that including parents, KA17, 18 are yellow Katrai type. So last year we bagged branches, we self-pollinated all the flowers on the single branch, many branch from at least about 3 individual from the F1 generation or children generation. And then we collected seeds and plant them. We already had about 1,100 plants already germinated, and we think we have about another 1,000 coming. So this is what that 1,100 germinated seedling looked like last month, which is about 5 months after we planted a seed. And this is a close-up look at them. Now I'm going to go back to the F2 or grandchildren population I mentioned. So these group of plants were planted in the field. They're about 20, 30 years old. No, 20 years old, 25 years old. And they were originally planted for DNA genomic mapping of coffee. But we're going to use this as tools to prove our concept. So, for example, we sent about 60 individual of these plants for DNA typing. And as you can see, they carry different proportion of DNA marker that came from the green bar is from Katimora 5175. And the red is indicate the DNA marker from the mocha hybrid. So they carry different proportion of marker from the 2 original parents. We also send about 60 plants to Lisa's lab for bioassay to identify which one is resistant to CLR. So KO34 is a Typica and it's susceptible. We use it as a control. As you can see, it produces lots of yellow spore. Both Obata and Katimora are resistant, so they do not produce spore. MA2C is the one of the parent from that cross. There are lots of yellow spore. And Dr. Lisa Keats were able to identify some plants that never produce spores. Well, that's an encouraging sign. And this is a different way to look at it. The higher the bar means more of the leaf disc show spores, produce yellow spores. So the higher the bar means they're more susceptible. The lower the bar means they're less susceptible. And we're very, very interested in these ones that never show any yellow spore. And we were able to identify 14 trees from the 60 that we sent to be resistant to CLR. And this is a different proportion of marker come from the two parents, from the susceptible but good tasting parent versus cultivar, the resistant but bad tasting parent. Interestingly, 5 out of these 14 never even produced yellow spot. Now, sometimes the cultivar, the resistant parent, even though no yellow spores were produced, there are some spots on the leaf that turns yellow. And their 5 line never even produced a yellow spot. So that's quite interesting. And this is a comparison between the leaf size of the 14 CLR resistant plant. As you can see, compared with the two parents, some look more like the Tomoka parent, some look more like the cultivar parent. Okay. Also, we were able to collect sufficient cherry from 11 of those 14 plants. Again, the size and shape of cherry, some look more like the Tomoka parent, some look more like the cultivar parent. I put in the other cultivar and a red Katrai just for comparison. And Miss Julie Burden, who is a trained cupper, will cut these 11 selection compared with red Katrai for us. And hopefully we will be identify one resistant one that are the taste, the quality is sufficient enough to be considered as specialty coffee. Now I'm going to talk about the Obata crosses. So last year, we selected similar set of the Hawaiian commercials as the ones that Shifumi did for cultivar and crossed it with Obata. And that involved remove the anther of the flower buds. No, we had to do this before the flower opened. Remove the anther and dust the female organ with pollen from Obata. So the male parents are Obata, and the female parents were the Hawaiian commercial cultivars. We also included 3 different selection of Ethiopian semi-wild coffee. These were imported by Shifumi from Costa Rica because they carry nematode-resistance. This work was actually Dr. Sayaka Aoki's master thesis, which she challenged these 10 different accessions of Ethiopian semi-wild coffee and compared them with Kona typica and yellow Katrai with the nematode that was found in Kona. And the higher the bar means the nematode reproduce more in the root of those plants. These are potted plants. And as you can see, all 10 Ethiopian coffee have very little reproduction of the nematode. So Julie also already cut 3 of them ET52, which is here, 25B here, and 17. And they're pretty good as compared to SL28 that was grown in the exactly same field right next to this Ethiopian plant. I have to point out the environment in Kunia, it's not conducive to produce good quality coffee because it's very hot and dry and no shade. So, for example, Typica, I was told Typica coffee normally grown in Kunia score about 80 to 81. So we think the quality of these Ethiopian coffee are pretty good. So we're hopeful if we cross this with Obata, we can identify coffee leaf rust and nematode-resistant coffee lines, also with pretty good taste. So this is what all the Obata crosses are seedling look like. They were planted a little later than the last batch. In fact, some of the F2 plant are also at this stage. And this is a closer up look of them. So again, for all these plants, after they are screened for CLR resistance and DNA typed, we will send them into 4 different farms on 4 different islands for field trial. These farms are actually most of the parent line we use with so-called Hawaiian commercial cultivar were identified from these farms. And hopefully in a few years, we will be able to harvest enough cherry from the single tree and have them cupped and identify good quality coffee. And here are the people I like to acknowledge. Sayaka is the postdoc working on the project, and Shifumi Nagai is a coffee breeder from HARC, longtime coffee breeder, and the HARC team, both in the field and in the lab, as well as my collaborators. Thank you.